Hello everyone, we have seen several laboratory techniques and field techniques for the estimation of water content. Another important state variable for establishing soil water characteristic curve, which is a fundamental constitutive relationship for soils, for partly saturated soils, is matrix suction or total suction. Let us see the different techniques that are available for either to estimate or control the suction of the soil. First, the basic technique is hanging column method or negative column method. A pictorial or illustration of the hanging column is given here, where a soil sample is placed in a container. You have a porous stone which is attached here, and this is soil. So, now the soil, this whole setup is connected with a flexible pipe, and water level is maintained up to here. When you allow the equilibrium to take place at steady state, you would see that the soil is completely saturated. If you raise this level, then water will start flowing. So, now what if you bring this column down? For example, if the column is brought down to this level, so water level is here, new water level is here, or it could be further brought down to this, water level is here. So, what will happen to the soil? Soil, because of the capillary action, soil tries to keep the water within the soil pores. Depending on the air entry suction of the soil, water may remain in contact with the soil, and you may have completely filled tube. This U tube will be completely filled with water, and that is a state you may have. So, now if you observe the pressure in the water, that will be negative pressure, because if this is the midpoint or midsection of the soil, there is a negative head of minus h meters or millimeters is maintained in this particular column. Under this negative head, soil is completely saturated or some water content may exist. So, this is on a soil water characteristic curve plot. If I plot the water content, either gravimetric or volumetric water content, this is negative head, or if I multiply with gamma W, this is pressure. I can represent in terms of pressure. When negative head is 0, the water content is fully saturated. This is Ws maybe, this is fully saturated water content. So, when the negative head started increasing, the water content may decrease, but still the soil may be saturated, because as I explained earlier, the meniscus curvature might change, but still water will be completely filled within the soil pores, and there is no air. So, it is completely saturated. However, water content may slightly decrease. As you increase the head further, you would suddenly see that there is a discontinuity between soil and then the water, and water is up to only here. So, that is a point where the air enters into the soil system. When air enters, there is a discontinuity between soil and then this water column. So, this is the simple way of explaining the hanging column technique, where how the water content changes with negative head. A very good soil water characteristic curve could be estimated using hanging column technique in this procedure also. In this particular procedure or method, we have a soil column. This is the soil, and which is connected to a water reservoir, and water level is here. So, water level in the soil is also here. You do not need to understand this particular thing as of now, but there are volumetric water content sensors are placed at different levels. So, these are EC5 sensors. These are EC5 sensors are placed at different levels, which can directly measure the volumetric water content, and these sensors are connected to the data logger. So, if you have such a system in place, now when you maintain the water content within the soil to this particular level, you would see that there is some water content that exists above this level also because of the capillary action. For example, at this particular height above this water level, there may be water content theta 1 and here at this level the water content is theta 1, here water content is theta 2, here water content is theta 3, here the water content is theta 4, here it is theta 5, here it is theta 6, here it is theta 7. So, you have around 7 sensors placed at different depth level. In this situation, now if I want to plot the soil water characteristic curve, on x axis I plot head and y axis theta. This head is negative because we are measuring above the water level. Our water head is negative above the water level. As we have seen in the capillary, if I draw the capillary here, which is immersed in a water reservoir, this is water, you would see that there is a water level that exists up to certain level. If you draw the pressure diagram with the depth, you would see that there is a negative pressure in the water in the column above this level, and here it is 0 and this is positive, this is negative. Similarly, above this you have a negative pressure. So, here the head is some value, correspondingly the water content may be very high, head is 0, the water content is theta s. And similarly, you have theta 1, theta 2, theta 3, likewise you can plot a soil water characteristic curve. In this particular case, you will have theta not significantly changing up to certain depth, but the soil is fully saturated, and beyond that you have water content decreasing drastically. Up to that point, or that head is called air entry value or air entry suction head. So, that is somewhere here. Beyond that you have partly saturated. You may have fully saturated system up to certain depth, that is HAEV, air entry value head. This is a actual setup that is available at IIT Guwahati Geotech lab. So, this is the soil column, and there is a reservoir, water reservoir, and several sensors are placed at different depths. They are connected to the data loggers. These two white colored boxes are data loggers. They are EM50, which are supplied by Decagon. So, here you get several data points. Similarly, now if I raise this water level to again certain depth, now this sensor realizes that the water content at this particular depth and at this particular depth both are theta s only. Now, this theta 3 would change and theta 4 would change. Now, you need to estimate what is a head from this particular point onwards. You get new points in between again. This way, you can establish the entire soil water characteristic curve for coarse grained soils like sands, because generally in coarse grained soils, because the pore diameter is smaller, so the air entry value is smaller. Also, the total capillary effect that is considered or that is available within the soil mass will be less. You can conduct such tests in the laboratory. In coarse grained soils, this is possible. Otherwise, if you have a fine grained soil, when you compact, as we have seen that the capillary raise takes place to very high values of height. So, therefore, it is not possible to conduct the tests by considering such columns. So, this hanging column method is a wonderful technique for establishing soil water characteristic curve accurately for coarse grained soils. So, here whatever is described here is a wetting technique, where the path is from right hand side to left hand side, because soil initially is a dry straight. As the head is decreased by raising the water level, the water content increased. So, this follows a wetting path. One can conduct the drying technique also. One can establish the drying path also by lowering the water level from the initial depth. For example, the water level is slowly raised up to here, and maybe it reached to maximum value. Now the soil is completely saturated and you obtain this particular point. Now when you lower down this value, this particular value to here somewhere, you can measure the water contents at different points. These points represents the drying path. So as the drying path lies above the wetting path, you would get the hysteresis can be established using this particular technique. So the hysteresis loop can be established using a single technique, that is hanging column technique, on the same soil sample without considering duplicate samples. However, you cannot have a very lengthy columns like beyond 1 meter, 2 meters. It is not possible to conduct the tests and not possible to have so many number of water content measuring sensors at different depths. This particular test is limited for coarse sands and maybe for fine sands. Generally, this is limited for coarse grained soils. A very common method that is used in unsaturated soil mechanics and also in soil science is tensiometer for measuring the suction value. This is a popular technique for measuring the suction in both science and engineering disciplines. And also this particular technique could be used in the laboratory as well as in the field. Earlier technique which we discussed, that is hanging column method, cannot be used in the field, which is purely a laboratory technique. When you alter the density of the soil, you can alter because it is a reconstituted sample. So then you get different soil water characteristic curves. But it is not possible to conduct this particular hanging column technique in the field. But however, this tensiometer is very advantageous because this can be carried to the field and tests can be conducted and directly it can be measured, suction can be measured. Now, so let us understand the principle behind this tensiometer. So, this tensiometer has a sensing probe and which is connected to a water filled body tube. So, here the body contains the water fully and de-aired water, and there should not be any vapor or air component in this, and de-aired water should be filled completely in this this body, this tube. Then it is connected to a sensing probe. This is sensing probe, and I will discuss what exactly the sensing probe contains. And this water filled body tube is connected to a gauge, pressure gauge. So essentially, this has a service cap which can be opened up to fill the water, de-aired water, and which can be closed during the measurement. Now, this sensing probe, when it is in contact with a water which is at negative pressure, the negative pressure is transmitted through this tube, and negative pressure is transmitted to the water which is contained in this body tube, water filled body tube. So, that can be measured using pressure gauge. So, what does it contain? The sensing tube contains a high air entry ceramic disc, high air entry ceramic cup which has uniform pores. So, these are all pores with a pore diameter of or pore radius of r, and this is a surface. This is surface and these are pores. Now, a uniform capillaries that contain in your sensing probe. So, as you fill this whole tube, so this is initially saturated completely. The sensing probe should be completely saturated. So, the capillaries are completely filled with water. That is the initial condition. This is a tensiometer, T8 tensiometer supplied by Decagon, which is available at our IIT Guwahati lab, Geotech lab. Now, this tensiometer, when it is in contact with soil, when the soil is fully saturated, the negative pore water pressure within the soil mass is 0. Soil pore water may be at atmospheric pressure. So, then water in the water filled body is also at the atmospheric pressure, then there is no issue. But as when the soil is partly saturated, so then there is a negative pore water pressure that exists within the soil. So, when that soil comes in contact with the higher entry disc, For example, the soil is placed here. This is soil, contains several grains, and within this there is a water. When this is in contact with this with these capillaries, air cannot enter into this provided the air entry suction of this pore space is higher than the air pressure that exists within the soil mass. Earlier, using Young-Laplace equation, we have estimated what is the air entry value. So, which is simply 2 Ts by R. If you know the radius of the high air entry disk, so you can estimate what could be the UA minus UW that could be sustained. So, this is the pressure across the interface that can be sustained. So, assume that this pressure that can be sustained here is 100 kilopascal. So, then when the soil contains negative pore water pressure, say up to 100 kilopascal, so air cannot enter, but water within the soil pores can establish good contact with the water in the higher entry disk, which is again connected to the water filled body. So, therefore, negative pore water pressure is transmitted through the water in the higher entry disk pores and is communicated to the gauge. So, gauge reads that this much of suction is existing because there is a negative water pressure. So, when it is transmitted, the water is sucked from the water filled tube. So, that much pressure that can actually detect and it can be read on the gauge pressure. So, when it is in contact with soil here, the pore water which exists in the soil is in good contact with the higher entry disk, and the water in the higher entry disk is in contact with the water through this and water in this tube. So, therefore, this negative pore water pressure in this particular negative pore water pressure, that is UA minus UW positive value, indicate that there is a negative pore water pressure in the soil. So, therefore, there is a suction or suction pressure that is exerted on the water which is available here. That pressure can be recorded on the pressure gauge. This way, you can establish the soil water characteristic curve, or for example, you take a soil column and in which the tensiometer is placed. So, this is the tensiometer and this is the soil. So, when the soil gets dried with time, directly the reading of the suction value can be monitored or measured. If you place tensiometer along with the EC5 or the volumetric water content sensors, both water content and suction using tensiometer both can be measured simultaneously at different equilibrium water contents within the soil mass, and one can establish the relation between theta and psi. So, similarly, therefore, it can be used for laboratory or it could be used in the field. So, that is the advantage of this particular technique. As we have seen, the limitation of this particular technique is the air entry disk, because if the soil contains a negative pore water pressure of more than 100 kilopascal or minus 500 kilopascal, but the air entry value of the high air entry disk is only 100 kilopascal. Water enters through the higher entry disk pores and water enters into this body. When there is a negative pressure within the soil mass which is in contact with the higher entry disk, the negative pressure may be as high as 1000 kilopascal. So, in that particular case, if the high air entry disk capacity is only 100 kilopascal, then the air enters into the higher entry disk, because the sustainable air water interface, sustainable pressure at the air water interface is only up to 100 kilopascal. There is a capacity of the higher entry disk. So, therefore, the air enters into the higher entry disk and air enters into your system. So, your pressure gauge will not read the values. So, therefore, the maximum value that can be measured by the tensiometer is dictated by the higher entry disk capacity. You have several higher entry disks. the higher entry disc, which is ceramic disc, the capacity, HAE capacity, are expressed in bar. 1 bar is atmospheric pressure. So, if it is a half bar, that ceramic disc will have a pore diameter of in mm approximately 6 into 10 power minus 3 millimeter, which is equivalent to the air entry value of in kilopascal. That can be estimated using Young-Laplace equation as we have done earlier. U minus UW is 2 Ts by R, 2 into its standard temperature. You may use 72.75 millinewton. If I represent in kilonewton, then it is minus 6 kilonewton per meter divided by the radius is 6 by 2. So, 3 times 10 power minus 3 mm. If it is represented in meters, then this is 10 power minus 6. So, 10 power minus 6, 10 power minus 6 gets cancelled. So, this is equal to about 48.5 kilopascal. So, the air entry of that particular disc is, it may vary around 48 to 50 or 55 kilopascal. So, that is why it is called half bar. Half bar is around 50 kilopascal. Similarly, if it is 1 bar, so the 1 bar higher entry capacity will have the pore diameter approximately 1.7 times 10 power minus 3. Here, however, in the calculation we did not use the contact angle. We are assuming the contact angle is 0. So, with that approximation, this comes to be around 48.5 kilopascal. For 1 bar, this pore diameter comes out to be 1.7 times 10 power minus 3, and 2 bar 1.1 into 10 power minus 3, and 3 bar 0.7 into 10 power minus 3 millimeter, so on and so forth. So, one can choose, and you have up to 5 bar and 15 bar as well. So, 15 bar will have the pore diameter of 0.16 times 10 power minus 3 millimeter. So, this is very fine. So, one can choose what ceramic disc we can use in tensiometer. One can use 15 bar, or one can use 5 bar, one can use 1 bar. But this higher entry disc pore diameter is not the only parameter that controls the maximum measurable suction in tensiometer. There is another important factor. If we revise our knowledge on phase diagram of pure water, so this is the diagram which I got for you. So, here with respect to temperature on x axis and pressure on y axis, if you plot, so this is the phase, this is the phase diagram. It represents several phases. This is the water phase, this is the vapor phase, and this is the water phase. We have seen that there is a cavitation that takes place when the absolute pressure decreases for the same temperature. At the same temperature, pardon about the scaling here, this is not a linear scale, this is some scale we have used here, and similarly y axis some scale we have used. So, here at the same temperature, for example, at say 25 degrees, when you are decreasing the absolute pressure of water, when it reaches the vapor pressure, this is the vaporization curve, this is the vaporization curve. If it touches the vaporization curve, or if it reaches the saturated vapor pressure, then water turns to vapor. So, therefore, how this is useful for tensiometer? So, in tensiometer, we have seen that there is a water filled tube which has a cap, and here this is connected to a gauge, and here you have vent, etc. So this is connected to one flexible tube through which you have a sensor which is connected. This is a sensor. This is a higher entry disc which is connected. Now with this sensor, when it comes in contact with soil, soil has negative pressure. So water pressure is negative within the soil mass. So U minus UW is positive. That means over water pressure is negative, right? So because UA is, if you take 0 as a gauge pressure, then UW is negative. Otherwise, if you take absolute, this is 101.325 kilopascal, and then UW may be some value, so then you have a positive value, right? You have some suction, so there is a negative pore water pressure. So now, due to this negative pore water pressure which is transmitted through this tube, and the pressure is transmitted to this water filled body, which is read on the pressure gauge here. Now imagine the water pressure within this tube is decreasing, because the soil is in contact with this, and the soil which is, this is the soil which is getting dried with time. So therefore the negative pore water pressure is increasing with time, which is transmitted through the tube, and the water pressure in this one is changing. So if it is changing, so if you consider the absolute water pressure PW within this tube, which is decreasing with time, initially it was at atmospheric pressure, which is equal to atmospheric pressure, which is decreasing continuously. When it reaches, so vapor pressure maybe at 3.1625 kilopascal at standard temperature, this is a saturated vapor pressure, UV sat, and depending on the RH, you may have some UV value slightly less than or significantly less than 3.1625 kilopascal at standard temperature. So when the pressure, water pressure reaches this particular value, then vaporization occurs. So this water turns to vapor. So you will see a lot of vapor bubbles inside the tube. So when the vapor bubbles form within this tube, then you cannot measure, because water volume does not significantly change. There is a pressure that is exerted on the water, that is you are able to measure. But when there is a vapors or bubbles that are formed within this system, then the measurement unit will not work, because there is a discontinuity that has happened here because of the discontinuity of water. So because of which it does not measure the negative pore water pressure. Actually this vapor pressure that controls the maximum measurable suction by the tensiometer. If the atmospheric pressure is say 101.325 kilopascal, if it is decreased continuously to the vapor pressure, so vapor pressure value, this is atmospheric pressure UA, this difference controls what is the maximum measurable range of your tensiometer, suction measurable range of your tensiometer. So for example, the atmospheric pressure is UA and the vapor pressure is UV. The difference between these two quantities would dictate what is the measurable range of suction of your tensiometer. So for example, at sea level, this UA value may be 101.325 kilopascal, and this UV, if it is 100% RH, UV is equals to UV sat at standard temperature, this value may be 3.16 kilopascal. This difference, whatever the value, maybe 98 kilopascal, is the maximum measurable value by your tensiometer. Beyond that, vaporization takes place and tensiometer does not work. Similarly, if you take the tensiometer to hillside, maybe 1500 to 2000 meters above the mean sea level, then the atmospheric pressure drastically drops. Atmospheric pressure may become maybe 80 kilopascal. Your vapor pressure at that point, maybe that can be estimated depending on the temperature. One can estimate maybe it may vary in the range of 5 or something, then the measurable range will become 75 kilopascal only. So, as your measurement is done at elevated places, the measurable range of tensiometer will decrease. This is at elevated places, this is at mean sea level, this is at mean sea level. So, we have seen the negative column technique or hanging column technique, where you can probably measure maximum value of about maximum suction range. You can establish, maybe if you have a 1 meter high column, then you can establish the soil water characteristic of up to 10 kilopascal suction. Beyond that it is not possible, then you should have increased the height of the columns, but it is not possible to have whatever the length of the column you want to have. Therefore, it has severe limitations in establishing the soil water characteristic curve for other than coarse grained soils. Coarse grained soils, generally the air entry value lies in the range of 1 kilopascal to maybe 3 kilopascal at max for fine sands. So, beyond that immediately the water content decreases as the suction increases. So, within 10 kilopascal range you can establish the entire soil water characteristic curve. So, for red soils or other soils where the water content significantly varies between suction range of 0 to 150 200 kilopascal, you cannot use hanging column technique, but you can use tensiometer up to the atmospheric pressure minus vapor pressure. In that particular range, one can use tensiometer to establish the soil water characteristic curve. You have another technique that is developed taking the inspiration from tensiometer. The technique is called axis translation technique. Actually, the name itself indicates the principle behind the technique. In this axis translation technique, you have a higher entry disc similar to the tensiometer, and in a closed chamber, thick vessels you should have in a closed chamber where one top is connected to the air pressure valve and another one is connected to the water pressure valve at the bottom. So, here the axis is translated, as we have seen earlier in the tensiometer, that as the elevation increases, atmospheric pressure decreases, because decrease in the atmospheric pressure causes the decrease in the maximum measurable suction value by tensiometer. What if you increase the atmospheric pressure? So, that is the principle behind axis translation technique. Air pressure is maintained at elevated values, maybe as high as you can maintain very high values, then water pressure can be maintained at lower values, maybe at atmospheric pressure itself. So, then if you see your UA minus UW, UA is positive because you are maintaining very high values, maybe 500 kilopascal. At air pressure you are maintaining, and UW is maybe 0 or which is equal to atmospheric pressure at gauge values. If you consider, a suction of 500 kilopascal could be applied without causing any vaporization, without causing any cavitation. So, therefore, that is the principle here it is used when the water level is up to here. So, the air entry disc is completely saturated and these are the pores that are formed, and the curvature indicates the pressure drop across the air water interface. On this air entry disc, if you place some soil like this, now soil pore water is in very good contact with the air entry disc. So, therefore, the water in the soil pore and water in the higher entry disc both are in good contact, they form a good channel. Now, when you start increasing the air pressure, so UW is constant, maintained constant, maybe equal to 0. So, then you are increasing UA, which is a positive value, that means this indicates the suction, matrix suction. So, you are actually maintaining certain suction. So, then Slowly the water, as UA minus UW is increasing, water starts bleeding from the soil, because water leaves from the soil, because you have increased the suction. As there is a good contact between a higher entry disc and soil, the water leaves through this and water comes out. So you can collect this particular water. If you connect to a burette stand, you may collect the amount of water that is coming out. So as and when UA minus UW is increased, the amount of water that is coming out is known. So when U minus UW is increased, which is plotted on the x-axis, U minus UW is increased, the water content, it could be gravimetric or volumetric, or here you are measuring the volume of water which is coming out, so it could be volumetric water content, which starts decreasing. For example, initially it has certain water content, and then you have placed inside, then you have increased a certain UA value and UW is 0, then a 5 kilopascal of air pressure is applied, then you have water content slightly decreased because some amount of water that has come out. Then at equilibrium, once the water stopped coming out, that means it has reached, soil reached equilibrium with the UA minus UW or atmospheric conditions, surrounding ambient conditions. Then you increase further value and some more water comes out. So this is how you can continue the experiment, just like applying a suction value, which is similar to your consolidation test. In consolidation, you have sigma applied and you have void ratio on the y axis. So this void ratio is similar to your theta. This is water content and this is void ratio. Both are state variables. Here UA minus UW, that is a negative suction or matrix suction, and sigma is a positive load that is applied on the soil. At equilibrium, where 100% or 90% consolidation takes place, this sigma is equal to sigma dash, then the curve is similar. The curve is, this is similar to this. Similarly, here also you are applying some load, matrix suction load. When you have increased, then water comes out, so the water content drops. Same thing is happening. However, the difference is the consolidation is conducted at fully saturated state. Here at any given point, the soil is at saturated condition, but here it is unsaturated case. Beyond air entry, there is unsaturation in the soil. So both are similar, but here air pressure is applied. Instead of directly measuring, we are controlling the suction. So this is not a measurement, this is a controlling suction. We control the suction and measure its corresponding water content to establish the soil water characteristic curve. So this is again a laboratory technique. This cannot be used in the field. So to understand the field dynamics, so you may have to get a representative soil sample and then place it in the chamber and conduct the test by increasing the air pressure, and this way you can establish the entire soil water characteristic curve. Here in this particular technique also you can do both drying and wetting, so you can establish the hysteresis. Here air pressure you are increasing, once it reaches certain value you can also start decreasing the air pressure and water will be sucked inside and water content will increase within the soil mass. So you will get wetting path. Here if the air pressure is beyond the air entry suction value of the higher entry disc, then water enters into the higher entry disc, and then therefore when this becomes unsaturated, when the air enters into the higher entry disc, so there will be a vapor bubbles that exists here, because air bubbles will start going into the water phase, then there is a discontinuity, you cannot measure beyond that. So therefore the maximum measurable suction range in this particular technique is limited by the higher entry disc capacity. One can use 5 bar or 15 bar, they are available in the market. So based on that, one can establish the soil water characteristic curve using this particular axis translation technique. The chamber should be strong enough because you are applying a huge air pressure. So you consider a thick chamber. So this is another technique which is used for establishing the soil water characteristic curve of laboratory techniques, which is called osmotic technique. Osmotic technique, which is based on the osmosis. Just recapitulate what exactly this osmosis is. When you take a U-shaped tube, when you pour water, both are at the same level, and also when you place a semi-permeable membrane, still both will be at the same level. Remember that the semi-permeable membrane does not allow the solute to pass through. Here water is solvent and solute we haven't added. So, therefore, water simply passes through from one side to another. So, therefore, it maintains the same level as it was earlier. So, in the second case, I have the semi-permeable membrane, but I have added some salts on the right hand side or some solute on the right hand side. As the solute is filtered, which cannot pass through the semi-permeable membrane, there is a higher concentration of solute on the right hand side, and here the solute concentration is 0 on the left hand side. This is on the right hand side. So, this is the condition. So, this is chemically in equilibrium. So, because the chemical potential of the right hand side system is lower than the chemical potential of the right hand side. So, generally the solute should have moved from right to left to make the equilibrium chemical potential throughout the system. However, because semi-permeable membrane is acting, semi-permeable membrane is resisting any movement of solute, it is filtering the movement of solute. Now, the water moves from left to right to dilute the solution that is available on the right hand side, which causes, this is the initial level of the water. Now, there is a decrease in the level here, and then that water is going to the right hand side. So, there is an increase in the head. This head is osmotic head, we have seen earlier. This could be HO, osmotic head. The same technique is used to establish the soil water characteristic curve. So, here you have a soil sample taken in a small column, and you put a semi-permeable membrane on the top and bottom, or only just at the bottom, and you have a porous stone which is placed. So, you have a membrane here, and this is a porous stone. So, now this is everything is contained in a column. So, this is the soil. So, now this is connected to a reservoir. If you maintain some solute or some solution, something like PEG solution, polyethylene glycol solution, which is maintained on in the column in the right hand side, the PEG molecule size is much larger than the filter size or the pore size of the semi-permeable membrane. There is a semi-permeable membrane. The semi-permeable membrane pore size is much smaller than the PEG molecule size. So, PEG molecules will be filtered to pass through. So, initially if the soil is completely saturated, then water will be taken out from the soil to dilute the PEG solution to maintain the chemical equilibrium. So, therefore, with time you see that the PEG solution concentration starts decreasing. The concentration of the PEG solution, initially C by C naught if you consider, initially it is 1, it starts decreasing with time and it reaches one standard value, one equilibrium, one steady state value. This concentration corresponding to one particular suction, and this concentration or this C by C naught value corresponds to what suction can be established based on PEG concentration. One can establish the PEG concentration versus the suction. So, based on this you have one particular PEG concentration, based corresponding suction value can be read, and that could be used. And this soil could be weighed, this particular soil could be weighed to measure its water content. So, one can establish water content versus suction. So, at equilibrium the suction in the PEG solution will be same as the suction in the soil pore water. at equilibrium. So, therefore, measuring the PEG solution concentration as well as the suction or chemical potential would help in establishing the suction value within the soil mass. Here also we are actually controlling the suction, we are not measuring the suction. By changing the PEG solution concentration, we are measuring the suction value. By measuring the suction value and weighing the soil, we get the water content versus suction that could be established. And but this suction is again total suction. In tensiometer, you have a higher entry disk and axis translation techniques. You have a high air entry disk. Disk pore size is not much larger than the solute molecular size. Therefore, it does not filter the molecules of the salt. So, therefore, you measure only the matrix suction there. It cannot curtail the movement of molecules. So, you will not get the osmotic suction. In this case, you are getting the total suction values. Whether it is a total suction or matrix suction, it only matters when the soil pore water consists of some salts. In case the semi-permeable membrane also arrests the movement of salts to either leave or to come in, then it only measures the osmotic suction, it does not measure the total suction. We need to be little careful about it. Otherwise, if there is no salts in your pore water, then this is equivalent to your matrix suction itself. So, then you have a suction versus water content relationship. That is the soil water characteristic curve. So, the soil water characteristic curve could be established. Thank you.